All right, so in this next video, we're going to talk a little bit about systematics and phylogenetics and how that's changed how we classify living things. So for about 2,000 years ago, Aristotle was kind of the first person who tried to divide living things into groups and classify them. He had animals and plants. He classified the animals based on where they lived, water, land, or air, and plants on size, short, medium, and tall. So the basic units of life are called genera. In 1750s, Linnaeus came up with his taxonomy scheme, and part of that was the scientific name, or this two-part name, or what we call a binomial name for all living things. So taxonomy is the science of classifying living things. A classification level is called a taxon. So we use scientific names because they don't change. If you talk about a bear, it could be a polar bear, a grizzly bear, a black bear. But if we use the scientific name, everybody that studies that organism knows what we're talking about. Whether they're speaking English, because they're Latin terms, they're the same in a language. And so they also don't change because Latin is not one of those, the meaning of words in English has changed over time. Because Latin's not really spoken anymore, meanings don't change. So here's the classification scheme. Domain is the most inclusive group. There are two domains, prokarya, or three domains, excuse me. The bacteria, the eubacteria, and the eukarya. Kingdoms, phylum, classes, order, family, genus, species. So genus and species are the two parts we use for the scientific name. So oftentimes we use mnemonics to remember things in order. The one I use is Dear King Philip came over for good spaghetti. So here you can see some examples. So there's been a lot of discussion about whether viruses are living things or not, whether they should be part of the classification. So they're not truly organisms because they don't have all the characteristics of life, so they're not in any of the kingdoms. They are parasitic chemicals, DNA or RNA wrapped in a protein. They can only reproduce in living things. They don't really grow, and so on. So because of the limitations of the hierarchy, the groups are not all equal. We've started looking at this hierarchical classification and looking at it in how it relates to molecular analysis, DNA, proteins, and so forth. So Linnaeus's taxonomy didn't take into account evolutionary relationships. Linnaeus's ranks are not equivalent. For instance, the legume family has 16,000 species, whereas the cat family only has 36 species. So we have this new classification system, phylogenetic systematics. So Carl Woese developed a six-kingdom system where he split the bacteria into the bacteria and the archaeobacteria. And then we have the protists, the plants, the fungi, and the animals. So the bacteria and the archaeobacteria were the two kingdoms of prokaryotes, and everything else is a eukaryote. So we're looking at this three-domain system, archaea, bacteria, eukarya. It's based on ribosomal RNA. Each of these domains form what's called a clade or an evolutionary group. The archaea and eukarya are more closely related to each other than the bacteria because they have more similar RNA structure, or ribosome structure. So since the fossil record is not complete, scientists rely on other types of evidence to establish the best evolutionary relationships. The study of these evolutionary relationships is called systematics. Phylogeny is creating a hypothesis about the pattern of relationships among species. So Darwin, we know, envisioned all species descending from a single common ancestor, depicted this history of life as a branching tree, and today we call that a cladogram, the branch diagram that shows the evolutionary history and relationships of organisms. The twigs represent the existing species. As you work back towards the trunk, you go back further and further in time. So again, Darwin called this process of evolution this way, descent with modification. So here's a couple phylogenetic trees looking at different possible relationships among these organisms. Again, the closer the nearest intersection point is, the more closely related those species are. So the key is looking at how recently species share a common ancestor. Just because things are similar, look the same, doesn't necessarily mean you have an relationship. You could have, convergent evolution and things going on that make things look like they might be related when they really aren't. So instead, we rely on derived characters, similarity that is inherited from the most recent common ancestor of an entire group. Ancestral characteristics are similar as arose prior to the common ancestor, further back in history. In cladistics, only shared derived characters are considered informative about evolutionary relationships. To use the cladistic method, character variations must be identified as ancestral or derived. If they're ancestral, we can ignore them. Derived, we got to look at them. So it can be morphology or structure, behavior, physiology, how they function, or DNA, RNA, proteins, molecular stuff. They should exist in easily recognizable character states. So to construct a cladogram, you have to polarize the characteristics. The clade are species that share a common ancestor, as indicated by the possession of a shared derived character. They all have the same shared derived characters. Clades are evolutionary units and refer to common ancestors and all of their descendants. Synaptomorphy, a derived character shared by clade members. So simple cladogram is a nested list of clades. The plesiomorphies are ancestral states. We don't worry too much about those. Symplesiomorphies, shared ancestral states. So here we've got a jawless fish, cartilaginous fish, a amphibian, a reptile, and 3 mammals. You can see the traits that were used to separate them out. The first one, jaws. The jawless fish doesn't have jaws, so he juts off. Everything else has a jaw. To the right of that, having lungs is where the salamander splits off from the shark. Being bipedal, having jaws, having lungs, amniotic membranes are all things we can use to separate out these species. Sometimes you have a shared character state that has not been inherited from a common ancestor. We call it homeoplasia. It's the result of convergent evolution. So it's kind of like the reverse of evolution, where things that had separated are coming back together or looking like they're coming back together. So anytime we have a conflict among characters, we use what's called the principle of parsimony, which basically means that the hypothesis that has the fewest assumption is probably the best one. So here we've got 2 different ones. You can see the one on the right has a few more assumptions than the one on the left, and so it's probably less correct. So how do we place species in higher groups into a taxonomic hierarchy? That's classification. So we can have what are called monophyletic and paraphyletic. Monophyletic groups are the most recent common ancestor of the group and all its descendants. Paraphyletic groups include the most recent common ancestor group, but not all of its descendants. So we've got 2 ideas about what is a species. We have the biological species concept, which has to do with being able to interbreed and produce fertile offspring. And the phylogenetic species concept, which is a newer one, focuses on shared derived characters. So species should be applied to groups of populations that have been evolving independently of other groups. Biological species concept cannot be applied to allopatric species populations where they are not separated. The phylogenetic species can't work in those cases. Phylogenetic can work for both sexual and asexual, whereas biological only works for sexual species. But because it's pretty new, the phylogenetic species concept, there's still some controversy around it.